In modern biopharmaceutical development, small and medium biotech enterprises face a silent regulatory adversity that can derail months of work, unexpected genotoxic impurities. A deficiency letter arriving 3 weeks before an IND submission or ANDA submission, citing incomplete nitrosamine documentation, can stall clinical programs indefinitely, triggering capital loss and delayed market entry. Regulatory uncertainty, analytical complexity, and timeline risk are the core obstacles, and even a minor omission can prompt a costly complete response later. Moving from regulatory vulnerability to clinical execution requires science-driven support. Resolve Mass Laboratory, an FDA-registered, ISO-certified contract research organization based in Canada, partners with drug sponsors to resolve complex impurities and build submission-ready dossiers addressing chemical mechanisms and regulatory gaps to safeguard timelines and patient safety. Analyzing the 5 costly nitrosamine assessment mistakes. A systematic review of regulatory deficiencies reveals 5 recurring failure modes in chemistry, manufacturing, and control documentations. These typically stem from incomplete understanding of organic chemistry, over-reliance on third-party documentation, or inadequate analytical methodology. Mistake number 1. Incomplete active pharmaceutical ingredient risk assessment. A primary failure is incomplete evaluation of active pharmaceutical ingredient synthetic pathway. Sponsors often limit assessments to the final synthesis step, missing precursor interactions in earlier stages. A compliant review maps every reagent, catalyst, starting material, and intermediate across the full route. Recovered or recycled solvents are also frequently ignored. Shared third-party recovery equipment introduces cross-contamination risk with secondary amines and nitrates. For example, recovered dimethylformamide can hydrolyze to dimethylamine, which reacts with trace nitrites to form NDMA. Sponsors must justify purge claims with empirical calculations or analytical verifications across crystallization, washing, and workup. Overlooking excipient-induced risk and material interactions. Sponsors frequently assume excipients are inert, a major regulatory gap since excipients are the primary source of trace nitrites and reactive amines in finished formulations. Providone K90, for instance, can contain diethanolamine up to 20 ppm and nitrites up to 8 ppm, enabling a solid-state nitrosation to an nitrosodiethanolamine over time. Relying on vendor certificates of analysis is a mistake. Suppliers rarely run high sensitivity nitrite or amine testing. Packaging matters too. Blister leading foils with nitrocellulose based primers can release nitrogen oxides during heat sealing that react with precursor amines on stability. Mistake number 3 is weak manufacturing process evaluation and parameter overlooks. The third failure mode is neglecting physical and chemical parameters during manufacturing. Hold times, water quality, and environmental conditions. Nitrosation kinetics are highly sensitive to pH and temperature. Under acidic conditions, nitrite ions from nitrous acid or dinitrogen trioxide, potent nitrosetting agents that react rapidly with secondary and tertiary amines. Below pH 5 and above 40 degree, yields can rise 12-fold versus neutral ambient baselines. Extended wet mass hold times during scale-up create a thermodynamic window for nitrosation, and teams often overlook processed water quality, ignoring environmental nitrate precursors under thermal stress. Quenching residual sodium azide with nitrous acid directly in the main reactor rather than segmented waste stream is an especially high-risk practice when precursor amines are present. Mistake number 4. Deficiencies in analytical strategy and method sensitivity limit. The fourth failure is an inappropriate analytical strategy. Methods that lack sensitivity and complete validation because acceptable daily intake thresholds sits in the parts per billion to parts per trillion range. Methods must achieve ultra trace limits of quantitation. A limits of quantitation above the regulatory threshold produces not detected reports that mask non-compliant concentrations. Sample preparation can also introduce artifacts. Acidic diluents or prolonged sonication can trigger in situ nitrosation in the vial, generating false positives. Matrix interference from polymers, excipients, or long-acting injectables suppresses ionization and skews recovery. Sponsors must validate specificity, accuracy, precision, robustness under ICHQ2 using isotope-labeled internal standards to correct for extraction losses and matrix effects. Mistake number 5 is unsupported qualitative conclusions and flawed exposure calculations. The fifth mistake is submitted qualitative low-risk declarations without empirical, analytical, or purge data. When an impurity is identified, sponsors often misapply toxicological exposure calculations. Less than lifetime thresholds can justify higher temporary limits for short-term or limited-run programs, but failing to account for cumulative exposure across cycles, defaulting to generic TD50 values when compound-specific data exists, or omitting uncertainty factors lead to rejection. A recent case illustrates the stakes. A long-acting injectable polymer formulation showed a nitrosamine drug substance-related impurity emerging near the regulatory limit during ICS stability. Trace nitrites in the custom PLGA had initiated a solid-state reaction with the API using high-resolution Q-exactive orbitrap mass spectrometry and optimized solvent extraction. The team isolated the impurity, proved the formation mechanisms, and established a control strategy, averting a projected 12-month delay. Mathematical treatment of exposure limits, less than lifetime calculations for products with restricted clinical exposures. Applying lifetime acceptable intake limits drives overly conservative actions, unnecessary recalls, or trial delays. Regulators permit less than lifetime calculations which extrapolate carcinogenic risk linearly from TD50 value. The chronic dose inducing tumors in 50% of test animals over the standard lifetime. The less than lifetime acceptable intake follows a typical formula. And regulatory expectations, what FDA reviewers look for in the CMC sections. A compliant nitrosamine control strategy belongs directly in the dossier's CMC sections, which is built on quality by design principles rather than end product testing alone. Reviewers expect a documented 3-step workflow in common technical document format. Step 1, risk evaluation, covering all ingredients, solvent recovery loops, process parameters, and packaging interactions. Step 2, confirmatory testing using fully validated GMP methods on multiple commercial scale batches and stability points. And step 3, risk mitigation, documenting controls preventing nitrosation, antioxidant scavengers, avoidance of nitrocellulose blisters components, and formulation pH held above the acidic threshold where nitrous acid really forms. Engineering a submission-ready assessment framework. To address all the challenges that has been mentioned before, developers should move from retrospective testing to a predictive life cycle monitored control strategy. The resolved mass formulation risk model predicts process-related nitrosamine risk during early formulation design, mapping critical quality attributes against process variables, and using reaction calorimetry and predictive thermal profiling to lock control points at GMP scale-up, engineering safety into process design, and streamlining approval. Conclusion and actionable recommendations. Rapid, uncompromised IND and ANDA approvals require sponsors to eliminate documentation gap, step-by-step evaluation of API synthetic routes, high sensitivity screening for excipients, derived nitrites, mapping of critical process parameters during scale-up, and analytical methods with LOQs well below regulatory thresholds. When selecting CRO or CDMO partners, project and regulatory leads should seek deep technical mastery, not transactional testing. Resolve Mass Laboratories deliver an in-house technical breach queue executive Orbitrap, GCMS, LCMS capability, custom synthesis of reference standards, and dedicated regulatory guidance, transforming compliance from a final checkpoint into a strategic clinical advantage. Thank you so much for watching this session until this, and I hope it helps you, and see you in the next one. Until then, goodbye for now.